Are you all now seeing the PPT?
Yes.
Yes.
So we'll start with the section 5, insects. What we are going to study over here, as I already told, it will be about insecticides, disinfection, the chemicals or the other methods that is being used, but specifically for the insects. And then how to control the insects in various other ways. The learning outcomes and the objective by the end, you will be knowing about the insects that causes various disease, because there is a separate section which you have already learned, the vector borne diseases. So there are so many vectors, especially the arthropod, that which we call as the insects, which carry the disease organisms, and its biting or stinging can cause diseases, various diseases for us. So vector-borne diseases are a very important, a very important public health important topic. So how to control them, insecticides, everything is so important. So insects, they are actually belonging to the phylum, the arthropod. And actually these arthropod comprise the most numerous and varied of the living things in the environment of man. Some of them are actually man's allies or actually friends, helping in fertilization of flowers because the insects like the bees, but vast majority are nuisance, even damaging the agricultural crops. Some of them even act as carriers or vectors of diseases. So the study that which we concentrate upon arthropods, especially the vectors or the insects which causes the diseases or the vectors which carry the disease causing agents. When we are studying that particular branch, we call it as medical entomology. So entomology, it is the study about the insects, and when we are studying not all the insects in the world, but only those insects which are very important medically, okay, that can cause diseases in human beings, that branch will be called as the medical entomology. So this topic, the insects, insecticides, the way to control the insects in medical, it is coming under the branch of medical entomology. So it is an important branch in the preventive medicine. So, what are the arthropods that we have to learn, which is of medical importance? Mainly, it belongs to the three classes: the class Insecta, the class Arachnida, and class Crustacea. So, in class Insecta, we have the mosquitoes, Anopheline culex mosquito or the culicines, and then we also have the Aedes and Mansonia. In case of flies, we have house flies, sand flies, sissy flies, black flies. Human lice, head and body lice, the pubic lice or the crab lice, fleas, the rat fleas, the sand fleas, the redwood bugs, and in arachnida, medically important will be ticks and also mites. In the ticks, we have hard ticks and soft ticks, and mites, leptotrombidium and trombiculid mites. And also there is itch mites, and also cyclops, which is belonging to crustacea. So these are the organisms which are important, that is belonging to the phylum arthropod. And what are the diseases caused by them? So we'll take the case of mosquitoes. All the mosquitoes we put together that we mentioned, the Anopheles, Aedes, Mansonia, and also Anopheles, Aedes, Mansonia, and Culex. So the diseases caused by them, they are malaria, filaria, viral encephalitis, like Japanese encephalitis, then West fever, viral fevers, then yellow fever, and dengue hemorrhagic fever. These are all caused by the mosquitoes. And housefly, mostly causes typhoid and paratyphoid fevers, diarrhea, dysentery, cholera, amoebiasis, poliomyelitis, even conjunctivitis. Remember the housefly causing conjunctivitis, trachoma, anthrax, and also yos. Sandfly, which causes kala azar. Sissy which causes the sleeping sickness. Lice or louse, which causes epidemic typhus, relapsing fever, trench fever. Rat flea, it causes plague and endemic typhus. One second.
Fantasy side, no.
Mm.
Then
No, Hanshipa,
black
dangerous.
fly, which causes onchocerciasis, redwood bugs, which causes Chagas disease, heart tick, tick typhus, tularemia, human babesiosis, and Casenor forest disease or KFD. Soft tick, which causes Q fever, relapsing fever, trombiculid mite, the strep typhus, and the rickettsial pox. Itch mite causing the scabies. Cyclops, which causes guinea worm disease, fish tapeworm disease, and cockroaches causing many enteric, they carry the enteric pathogens, so causing the GIT infections. So these are the most important arthropods belonging to the different classes. And what are the diseases caused by them? That is what we are going to learn. The diseases caused and its prevention will be leaving, studying in later topics. But today, in this topic, we are going to concentrate on how to control all these flies, these insects, and how to prevent the diseases by controlling them. So, as I already told you, this topic is mostly important in case of a examination point of view, not much in a practical aspect, like in a clinical aspect. Uh, and most of it, it won't be like when we go to the chapter, you will see how we are controlling. But for the exam purpose, it will be never asked like how you can control mosquitoes. The questions usually formulated will be asking about malaria or filaria, and then the question will be that towards the latter part of that question, the question will be how to control filariasis. So one of the method of controlling filariasis will be the control of the mosquito that is spreading, and then for that question, for that part of the question, you have to write the answer from here. So that is how the question will be formulated, and that's the importance of this chapter. So no particular questions. MCQs may be asked, but no particular question like how to control this particular mosquito. Never. It will be always associated with the disease. So initial part will be to explain about the disease, and then the latter part of that question will be how to control it. Then this chapter comes into use. So you should remember which are the organisms causing the particular disease. Suppose I may ask only onchocerciasis. So you need to know that it is caused by black fly and then how to control the black fly. So what are the basic principles of arthropod control? The general principles are actually four type of controlling we can do for the arthropods. The first one is environmental control. The second is chemical control. The third one, biological, and also the genetic control. So environmental control, this is actually the best idea and very inexpensive type of method and giving a long-lasting result also, almost permanent. So what in case of environmental control they do is like I mean, we can put that under the environmental sanitation because all these organisms or the arthropods, they're having a particular habitat in which they are breeding and they are growing up or developing. So in environmental control, what we actually do is to that source cutting or source reduction. That's the actual term that is being used. We are going to eliminate those breeding places. So that is one of the best approach, less expensive, and also the results are going to be permanent. That is the environmental control. So some examples are the best one, the source reduction, that is elimination of the breeding places. Filling and drainage operation, especially for mosquitoes. Out of all these arthropods that we have mentioned, the most universal one and the most menacing one will be of mosquitoes. So, the mosquitoes are usually breeding in a collection of water, and the different species that we told about, we'll see in later slides that all these species, they have special affinity for certain type of water for their breeding. We'll see in later slides. So we need to remove their breeding places, and also if there are some organisms or this not the organisms, sorry, the flies or the insects may stay live in the crevices of the walls, doors, etc. So better we close that crevices, the filling, the drainage option, if there is some water accumulated, to have good drainage operations. Then carefully plant water management, provision of pipe water supply, proper disposal of refuse, especially because it's in the garbage, the refuse, and other waste, the houseflies. The houseflies, their breeding place is actually this refuse, garbages, etc. So, to proper dispose that, to maintain cleanliness in and around houses. And for all these things, what we need is the cooperation of the people. If they are not keeping their house area clean, and mostly what we see is the people will might be keeping their house area clean, but not the nearby or the surroundings, not their immediate vicinity, but just after to the next house, they may be throwing the garbages from their house to the next ground that is present. So they will be keeping in their house area to be very clean, in a sanitary measure, everything will be done, but may not be in the neighborhood. They will be like, that's none of my business. But a cooperation is required, because mosquito, when it is breeding, it is not looking up in which household it is living, and then they are going to cause the disease only in that household. It's nothing like that. So we need to have a cooperation from all the people in that area. So the most ideal method to introduce the environmental control will be giving proper health education, and also political support is very important to make the necessary policies, etc. So that is about the environmental control. Then chemical control. So as the name indicates, in chemical control, we are going to use a lot of chemicals to kill the organisms. So we can use insecticides belonging to organochlorines, organophosphorus, and carbamate groups. In the later slides, we'll see what are the chemicals or the insecticides which are coming under organochlorine. DDT is most important coming under that. And you know DDT is a widely used insecticide. Then organophosphorus is there, and carbamate groups are there. So this can be used. That will be, because it is chemicals, it is actually a chemical control. So it must be mentioned that vector control by insecticides alone is no longer fully effective because of the growing resistance from these insects. So insecticide alone, another thing is they can cause the pollution also, the soil, the water pollution, and even the symbiotic organisms or the flies may be getting killed. So there is those disadvantages are there by using insecticides. And also another one is the organisms are becoming increasingly resistant to the insecticides. This, coupled with the danger of environmental contamination, had led to restricted use of insecticides in some of the countries. To avoid undue environmental pollution, it is now considered essential to replace gradually the highly persistent compounds such as DDT with compounds that are easily biodegradable and less toxic. So those chemicals will be methoxychlor, abate, and dersban. So earlier it was DDT that was widely being used, but DDT, it has got benefits and disadvantages also. It is having a long residual period, which means it will be leaving its presence or a air film over the surface for a longer time. So it is beneficial in some manner, because last class when we were doing, we learned about the criteria for an ideal insecticide, and we saw that one of the ideal criteria should be a long residual effect. So that makes DDT a better choice, but that itself could cause the pollution and the other related things. So nowadays, in case of DDT, they are or the scientists, they actually want to replace it with the chemicals which are less toxic, especially methoxychlor, abate, and dersban. As there is no alternate control method which is as efficient and economical as insecticides, it is postulated that most of the developing countries will have to depend for some time to come on the organochlorine compounds, because this is very efficient and economical. The environmental pollution is there, but still this is one of the ideal methods to be followed, at least in developing countries, for some time from now, unless they find out some other best methods. Then we then go for biological control. So biological control will be, as the term indicates, biological or some living organisms. Some living organisms are used for killing these vectors, especially the mosquitoes. And when I say like that, some of the thing, the most important thing that will come into your mind might be about the use of certain fishes. We call it as guppy, so or gambusia, fishes belonging to that species. So these are fishes which actually not breed, which actually feed on these mosquito larvae. So we can use those fishes to kill the these vectors. So to minimize the environmental pollution using these toxic chemicals, etc., we can go for the biological control. So we can use the larvivorous fish. Oh, these fishes, the gambusia, and there is a fungi of the genus Coelomyces, and then another one is guppy, which I told you. They can be used for mosquito control. A variety of other biological agents like bacteria, fungi, nematodes, protozoa, viruses, they are also under study for the control of insects. But there is a fear because if we are going to introduce biological agents for the control of arthropods, it is also possible that after some time, the new varieties that we are introducing to control the insects, these new varieties of bacteria or nematodes or whatever organisms they are going to use, they are actually going to become another important menace for the human health. So there is a risk of that, but still this is an ideal method. Uh, so here this is the gambusia or the mosquito fish, and then the fungi belonging to the Coelomyces, which can kill actually the mosquito larvae. Then the last one is genetic control. So nowadays, many studies are being done in genetic control, but if it is being developed and if we can do it in a high scale or a large scale manner, then it is very ideal, one of the best method to follow. But even at this stage, it is only in a developmental stage, like studies are being done only. So until this is properly developed, we have to rely on other methods, especially the environmental method and also the insecticides. So in case of genetic control, the main techniques that is being used is sterile male technique, cytoplasmic incompatibility, and chromosomal translocations. These are found to be effective in small field trials, but whether this can be used in a large scale or not, whether it will be feasible financially and other resource wise, we have to see in the future. So now we go into this technique, especially the sterile male technique. So you understand, what happens is they are taking those particular males or particularly breeding it, and then they are making the males to be sterile, the most male insects, and then leaving it into the environment, so that the population, the breeding, even if it happens, the population will be reducing because the males are being made sterile, the male insects. So that is sterile male technique. Another one is cytoplasmic incompatibility. And in cytoplasmic incompatibility, what happens is there is a particular bacteria which is called as whole bacteria. Wolbachia. Wolbachia. This is a particular bacteria, and it has been found that this bacteria, this Wolbachia bacteria, if it is infecting the mosquitoes, especially the male mosquitoes, and uh, that infected male mosquito, if it is going to mate with a female mosquito, because of the infection caused by this bacteria will be actually residing in the cytoplasm of the mosquito. The mosquito cell cytoplasm will be residing, and this bacteria will be changing, producing some kind of changes in the body of the male mosquito, and also the sperm of it. And as a result, what happens is when this infected male mosquito, when it is mating with a female mosquito, uh, because of the changes that has happened, this sperm will be killing the female eggs so that the mosquito will not be produced. So that is one of the method, that is the method used under cytoplasmic incompatibility. So when the scientists recognize that this particular bacteria, that is Wolbachia, do have this ability to change um, the genetic composition or the cytoplasmic composition in such a manner that the sperm of the infected male mosquito on mating can kill the eggs of the female mosquito. Now they are trying to do it in a large scale to see how whether this is a feasible method to control the mosquito population. So what I now mentioned is actually the second thing, where the infected male by this Wolbachia, if it is breeding with a normal mosquito, because of the changes that has happened in the sperm, they will be killing the eggs. So that because of that, the offsprings will not be produced. But there are other combinations also possible. You can see from the picture. So if it is happening in case of non-Wolbachia infected mosquitoes, so then there is offsprings that is going to be produced. And they have seen the combination if the female is actually infected. If the female is infected and male is normal, then also they produce the mosquitoes. But here it will be Wolbachia carrying offspring. So this is also useful because if those offspring they are getting naturally infected by the bacteria, then at least in the coming up generations, the population of the mosquito will be reduced. And then another combination is whether when it is happening between two infected mosquitoes, both male and female being infected, then also they produce this viable Wolbachia offspring of that mosquitoes. So all these methods, the three methods are ideal to do this technique of controlling the mosquito population. The best one will be where the male is actually infected. So the in the first generation itself, the sperm is killing the female mosquito eggs, so that the breeding is or the egg is not being formed or it is being killed. And the other two methods, we get the already infected mosquitoes, so we don't have to trap them and then do this infection artificially. It is being naturally grown, and in the second generation on, we can see that there is a reduction in the mosquito control. So this is one of the genetic control measure, the cytoplasmic incompatibility, in which they are using the bacteria and then changing the property. The other one is chromosomal translocation. So you know from the genetic chapter, chromosomal translocations. What is chromosomal translocation? Can anyone say me what is chromosomal translocation? Someone? What is chromosomal translocation? Seniors? Are you all there?
Yeah.
Okay. So someone, you have given me pages and pages of uh, assignment.
It's
Anyone
a
present? Huh.
it's a genome abnormality, is it? That
Yes, yes,
part of the chromosome breaks
yeah.
and then it reattached to a different chromosome,
Yes.
to a different location.
Yes, yes. So that is something naturally that is being happened. And here what they do is they're going to manipulate it. And mostly what they are going to do is rather than translocation, they are going to, or the genetic engineering methods, the scientists will be cutting a part of it, of the chromosome, and introduce new section. And this newly introduced genes or the chromosome, they have the ability to change the natural reproductive cycle of the organisms. So, and this is mainly done for mosquitoes like mosquitoes which spread the disease like malaria, Zika, and also dengue fever. So this chromosomal translocations, or rather introduction of genes into the genetically engineered genes into the mosquitoes, they are going to disrupt the normal reproductive cycle. So that is happening in chromosomal translocations. But when I say this, the methods, you can understand how much costly it will be as of now. And in a small scale, it seems to be feasible. But as we say here, whether it is feasible in a large scale, whether it would be within the budget of developing countries or not, it is to be seen in the future. But once we are able to do this, if these genetically engineered mosquitoes are not becoming another menace, then this is one of the best method. If it is not expensive, also we don't have to rely on the insecticides and then result in the environmental pollution. We can go for this, but there are limitations. So this is the genetic engineering, and some newer methods are the which is now being used for pest control, and that is insect growth regulators, chemostimulants, sex attractants, or pheromones. And from these terms, you will understand it is not genetically actually done. Some chemicals are used actually, some growth regulators, some chemicals are being used, the chemostimulants. So some chemicals are used as to induce the sterilization process, and pheromones are being used. So here the chemicals are being used so as to disrupt some of the process in the reproduction of the insects or the mosquitoes or anything. And this one is exactly, these three are exactly a manipulation in the genetic level. The manipulation is being done. So we'll see what is happening in case of insect growth regulators. They are actually certain chemicals that is being used, and what they use, what they do is they are not going to kill the insect per se, but they will be affecting uh, the ability to grow and mature normally. Because in case of this, as the figure explains, you can see there is the fleas, the sand fleas or the rat flea, and they will be going through the life cycle, the egg, the larva, the pupa, and then molting to the adult one. So if we are going to use this chemical, what they do is they are going to break that cycle. Like the flea eggs, it is not allowed to get converted to a flea larva. Or they may be breaking into another part. The pupa is not allowed to grow into a normal adult. So here the IGR, they are not killing the organisms specifically as like we do with the insecticide, but they are actually affecting its maturity. Or they are not allowing it to complete its growth cycle. And but they are actually chemicals. So this will be affecting the ability to grow and mature normally rather than killing them outright. Then chemostimulants are the, as I told, the certain chemicals are used to induce sterility in the vectors. And these chemicals can be either be given via food, or the entry to the insect can happen via mouth or just by contact. And they also actually disrupt the reproductive cycle because it's a sterilant. This is going to disrupt the reproductive cycle. And what they go, what when this is used, what is happening is this can affect the normal cell division that will be prevented. So the zygote to develop into a normal organism or the normal insect, it will be affected. Or it can inhibit ovarian growth and development. Changes will be induced in the structure of DNA and RNA, and that is going to disrupt the normal reproductive cycle. So chemicals that is used to induce sterilization in the insects, they are called as chemostimulants, introduced into their body via orally or by contact. And one of the example will be chloroethylamine. Then the last one is the pheromones or sex attractants. And now, from your school level knowledge, you will be knowing the pheromones are actually certain fragrance of chemicals that is produced in the insect's body, so that during the mating they can attract the males. It's actually produced in females actually. So during mating, it will be attracting the male insects. So this particular, that is what is happening in the natural manner. Scientists wanted to use it in a manipulative manner so that they produce these pheromones, artificial methods, and then this act as a trap. So when they use these pheromones and keep it in a closed enclosed area, the male insects will be coming over there. They get into that trap, and after they are getting trapped, either they can be sterilized or they can be prevented from mating, or whatever be the purpose, so that the population can be controlled. There is one advantage, because pheromones are actually species specific. So if you want to control a particular species of insects, this method is very ideal. Because the other methods, like especially the insecticides, as I already told when I was saying the insecticides, it's not only that particular organism or that insect being killed, even the symbiotic one or the ones that are useful for human beings, even that will be killed in case of insecticides or any pesticides. But when we are going to use pheromones, because this is species specific, and we are going to develop those pheromones, the male counterparts of that particular species only will be getting trapped, not all the insects. So that is one of the advantage of using the pheromones. There is a high level of specificity. So we need to target only the particular pest without affecting the non-target organisms, which means this is environmentally friendly. However, this is not totally effective or not a complete solution. We have to use other methods also. So these are the different methods. And so finally, we can say that there is no single method. We explained the environmental method. We said about the insecticides or the chemical method. We said about the biological method, the genetic control. But when we do each individually, we know only there is no one particular technique that which we can rely exclusively. We can never say that okay, we do the environmental control, then all the vectors or the insects can be controlled or can be killed. We can never say that. It should be actually a integration, two or three or all the methods coming together, or if not the genetic, the other methods coming together. So an integrated approach is always very necessary to control these insects. So since no single method is likely to provide a solution, we need to go for the integrated, where two or more methods can be combined together to get maximum results in minimum effort. So this is the basic principle of controlling the insects. The environmental method, where there is a source reduction, the breeding places are being eliminated. The second method being the chemicals, the organophosphorus, organochlorine, and carbamyl. Those insecticides are being used, but there is a risk of environmental pollution. And the third one, the biological, the use of certain fishes, certain fungi, and the last one being genetic, but it is still under research. So the ideal will be an integrated approach. Now going towards the insecticides. So insecticides, as the term indicates, it is going to kill the insects. And actually this is coming underneath the umbrella term pesticide. So pesticide is the vast umbrella term, a general term. And underneath these pesticides, we have the different types of sites that which kill each particular group of organism. So if it is killing the insects, insecticides. If it is killing the fungi, fungicides, rodents, rodenticides, herbs or herbicides, disinfectant, repellent, and all the other chemicals used for the control of pests, it will come under the umbrella term pesticides. And insecticide is one of the branch, one of the term within that umbrella term. So this control of arthropod disease, arthropod-borne diseases by insecticide is one of the greatest times of public health during the 20th century, because when we have learned those public health is history in epidemiology, we have seen that in earlier times it was actually the communicable diseases that was causing a menace, a high rate of mortality. Now later it was changed, there was a transition in which the communicable diseases mortality reduced and the mortality due to non-communicable diseases were increasing. Now it is like both are increasing, but still in the 20th century, that mortality and the morbidity caused by this vector-borne diseases or the arthropod diseases, that being controlled by insecticides, one of the greatest victory in the public health arena actually. So insecticides have not only controlled malaria, plague, typhus, and other diseases, but it's also in other manner useful because this will keep the people healthy. Their lifespan is increasing. Otherwise, when these communicable diseases were there, the people were dying at a very young age. So, by the use of these insecticides, these communicable diseases being controlled, environmental sanitation being maintained, the lifespan of the people also increased. They were more healthy, the mortality rate being decreased. So, that was being bringing, if the people are healthy, living for a longer time, of course, that is beneficial for the country because it brings about economic development and also social benefits because when we go for these pesticides or these insects, they can actually cause a lot of um, crop the agricultural crop destruction. So when they're controlled, it of course causes pollution, yes, but forget pollution for a second. It was actually helping the increasing the food production or the agricultural crop destruction were not happening with these insecticides. So it was not only the control of diseases that was happening, but there was social and economic benefits also by the use of insecticides. So how these insecticides are classified? Two types of classification can be seen. One is how they are going to act on the insects or how they are going to kill those insects, the mode of action. That is one classification. And second classification is based on the chemical, the nature of the chemical that is being present on them. So based on the mode of action, the first classification is the contact poison, the stomach poison, and fumigants. So contact poison, the name in itself indicate when these insecticides or the chemicals are coming into contact with uh, the organism, they are going to be get killed. So here it is due to contacts, and example will be pyrethrum, DDT, HCH, dieldrin. These are examples for contact poison. So DDT, it's mainly used like for the insects, but also for the ants. So when we put this DDT powder over to the ants, we can see they will be dying very soon actually. So it is by that contact when it is being put on their body, the poison being active. The stomach poisons. So stomach poisons, which means this poison should enter into the GIT or the stomach of the organism. Then only they are going to kill the organisms. Otherwise, just by contact, they are not going to kill. Somehow this chemical should reach their alimentary canal. So stomach poisons are those which when ingested cause the death of the insects. Examples will be Paris green, sodium fluoride, etc. And fumigants. Fumigants means fumigation or some air spraying. So here it will be giving off vapors, and when they are going to respire this aerosol or the vapor, that is going to produce a lethal effect on the insects. Example, sulfur dioxide. So in the mode of action, we have the insecticides classified into contact poison, stomach poison via GIT, and if it is fumigation of the respiratory tract, it will be the fumigants. Then based on the chemicals that is present, the group 1 is organochlorine compounds. So the main one coming under this organo. So organochlorine means the chlorine will be the most important ingredient in these chemicals. So the best example for organochlorine will be DDT, HCH, dieldrin, chlordane, methoxychlore, etc. Then group 2, that will be mostly with the phosphorus, the chemical. So the example will be malathion, phentheon, chlorpyrifos, abate, etc. And group 3 is carbamates, in which the examples will be propoxer and carbaryl. You can expect MCQ from this, like DDT being asked in which category it belongs to, or the other way around, one of the example of organochlorine compound. So some MCQ you can expect from this. You need to study only the main one actually. So this is a chart indicating the different classification of insecticides. I don't want you to by heart all these things, but at least the very important one you should be knowing. So you can see they have been classified mainly into the contact poison, the stomach poison, and fumigants. And from the stomach poison, remember Paris green. From the fumigants, remember hydrogen cyanide and also sulfur dioxide. In the contact poisons, we have naturally occurring and also synthetic. So naturally occurring, there are certain plants, especially pyrethrum, rotenone, nicotine, which are naturally occurring and can be used as a contact poison. And then there are chemicals which are synthetically developed. So in the contact poison study, pyrethrum, rotenone, and teres, it will be coming up in the later slides. So you can better remember, and in the synthetic, we have the organochlorine, organophosphorus, and the carbamates. In the organochlorine, just remember DDT, HCH, yeah, mostly dielrine. And in organophosphorus, the most important will be malathion, fenthion, and abate. Then carbamate, OMS 33. Yeah, that will do, not anything else. So we'll go into detail of certain important insecticides. Actually, in the textbook, detailed description of all the insecticides have been given, but I have selected only a few one that is very important for you to know. So we'll just go through that only, those insecticides or those chemicals only. So first one is DDT or dichloro diphenyl trichloroethane. Remember this chemical name of DDT, and this was first synthesized in 1874. But in the next slide, you will see that another one insecticide, HCH, it was discovered even before to DDT. But even now, it is the DDT that is more prevalent than any other insecticide. Though many of the organisms have grown resistant to it, that's also true. So DDT, as we already told, it's a contact poison. When the organism is coming into touch with this DDT, they are getting killed. So it actually acts on the nervous system, and how they are going to kill the insects will be they causes the paralysis of legs and wings, and then it will produce convulsion and finally death. That is how they are acting on the nervous system. But the DDT does not cause immediate death, but it takes several hours to kill. And this I was remembering that when I was a child, I used to put this DDT. It was very easily available at that time, but not that easily available nowadays. Uh, so from the packet, I will be directly putting it on that group of ants. And some ants used to die immediately. Some will be running off. And then I also used to run off behind this insect, the ants, and then put again, then it will be again running off. But yeah, because some may die immediately, but not all. It will take usually several hours or minutes to hours for the DDT to kill. However, the most important why it is being used is the long residual action. This long residual action is actually the advantage and disadvantage also. Advantage in that these protected surfaces will be having that residual effect for a longer time, so that that area will be free of these insecticides. We don't have to apply it frequently, but then it can even result in various types of pollution. So this long residual action of DDT is one of the reasons why it has been widely accepted in the earlier century, the 20th century. So it is about 18 months depending upon the treated surface. HCH, as I already told, this was discovered by Michael Faraday, not discovered, he synthesized actually, much earlier than DDT, that is by year 1825. And DDT was actually in 1874, right? 1874. So almost 50 years later, only DDT was discovered, not discovered, synthesized, sorry. So HCH or BHC, its full form will be benzene hexachloride or hexachlorocyclohexane or gamexane or hexitol. Try to remember at least why the term hexa or hexi. Try to remember because this may be asked as a MCQ. So this is more insecticidal than DDT. But the thing is, they're going to immediately kill the organism if HCH is being used. They are very much more insecticidal or more strong than DDT. But the only disadvantage in them is it is not having a long residual action. So the HCH kills insects by direct contact, as like DDT itself, but the residual action is only for a short duration, that is for only 3 months, when compared to the 18-month residual action of DDT. So that is why the BHC never became that much widely popular as like DDT. Then we have malathion. It's actually an organophosphorus. The other two that we discussed now, that is DDT and HCH, it was an organochlorine. And malathion and abate, it is actually organophosphorus. So malathion is actually least toxic of all organophosphorus compounds. And because of its low toxicity, it is considered as an alternative insecticide to DDT. Remember this, it can be asked as an MCQ. The other one is abate or temiphos. It is also an organophosphorus compound. It is of low toxicity, and this is used exclusively in India for the control of Anopheles mosquito in wells and also in domestic water containers, with at a dosage not greater than 1 parts per million. But this is more for a larvicide actually, to kill the larval, the mosquito larval. So this Anopheles is usually seen in the wells and also domestic water containers. So if we use it in the dosage, which is not greater than 1 parts per million, this is highly effective. But to kill the mosquito larvae only, this abate is not effective to kill the adult mosquito. And why I mention abate is, once in one residence that I was staying earlier over here in Malaysia, at that time, around about June, July, no, March, no, April, April, May, around that time, I think, April, May. At that time, there was increased dengue fever cases in that area in my residence. And I don't know whether it was routine over there, because after that I shifted from there. So people had come and to check each uh, residents individually to see the bathroom, and also this kitchen area, and to see whether the enough hygiene and sanitation is being maintained or not. And when they were leaving, they gave me some of the chemicals that could be used to put it in the accumulated water anywhere. And one of it, the packet, it showed abate, and abate was used because of its low toxicity, but it is useful only for as a larvicide, but not that much as an adulticide. Then we have diazinon. It is proved effective in the control of DDT resistant insects. So already we told, in case of DDT resistant or the less toxicity, we can use malathion as an alternative. But in those organisms where they have grown resistive to DDT, we can use another option as diazinon. It has given satisfactory control of flies and mosquitoes. However, it is more toxic than malathion or fenthion to man. So even though this is an alternative for DDT, the DDT resistant insects, even malathion, but when we consider the toxicity, malathion is less toxic than diazinon. So mostly it is the malathion that is being used rather than diazinon. Then fenthion, it is found as effective as DDT, and it is highly effective in controlling the larvae of Culex fatigans, mostly causing filariasis. So fenthion is mostly used to control the larvae of the Culex variety. Then dichlorobos, this has been successfully used for disinfecting aircraft. And then we have the natural ones. All these were the chemicals, the synthetic one. Now the insecticides which are developed naturally, and after seeing that this can be naturally, I mean naturally that it is being that is being present, now the scientists are actually synthetically producing it. So pyrethrum is one of such insecticide which is naturally developing, or it is extracted from the flowers of chrysanthemum Cineraria folium. And this is a plant which is cultivated in India in Kashmir, Shimla, and Nilgiris. So this is the plant, and they have that active principle, the pyrethrum, which can be used as a insecticide. So these all act as nerve poisons. But the main problem is with pyrethrum also there is immediate death of the insects. But the main problem over here also there is no residual action. So it does not possess the residual action of DDT. Most of the space spray, like outdoor spraying, etc., they actually use pyrethrum and DDT. So, because of the benefit, in case of pyrethrum, as I told you, there will be immediate killing. In case of DDT, there is no immediate killing. It's only taking place after a few hours or within a few hours. But in case of pyrethrum, there is no residual action, and in case of DDT, there is a long residual action. So they are complementing each other. So for that, in space sprays, they use a combination of pyrethrum and DDT uh, because so that we get a synergistic action. And now, because a large quantity of the plants have to be produced uh, uh, to extract this pyrethrum, they go for synthetic pyrethroids being developed to replace natural pyrethrins. And this synthetic one is found to be 10 times as effective as the natural occurring pyrethrin. The other naturally occurring one is rotenone, that is obtained from the roots of a plant, Deris elliptica. So this was widely once used for the control of flies, fleas, mites, and ticks. Not the mosquitoes proper, but the other kind of arthropods, the lice, fleas, mites, and ticks. Is also naturally occurring. So these are the most important insecticides that I selected for you people to study. So the classification, the two classifications of these insecticides, one is based on their mode of action, and the second one based on the chemicals that is being present. Then the important ones being DDT, HCH, malathion, fenthion, abate, dichlorovos, and pyrethrum, and another one being rotenone. Now we go towards the main issue, that is insecticide resistance, which is a very big problem. The widespread use of synthetic insecticides that has given rise to the serious problem. And the magnitude of problem is so big that we can understand it. In 1946, resistance to insecticides was only reported for 2 species of insects. In 1946, only 2 species were resistant to the insecticides. Remember, all of these began to be started using like DDT. It came, it was synthesized only in 1874, and from 1874 to 1946, how much years might be? 50, 25, around 75 years, around 70 years. So in that gap, it was only 2 species of insects which were resistant to the insecticides. But in 1962, like after maybe 15 years, yeah, 15 years, the number from 2, it rose to 81. And in the next 10, 15 years, 1980, this number, it rose to 134. And now I didn't get the recent one. Recent one will be more than this. So that shows the problem, the insecticide resistance, how vast it has become. And earlier, one of the benefit, as I told already in the earlier one, there was a food or the agriculture crops were not getting destroyed by the pest. There was benefit, but there are side effects of using this agricultural pesticides in a larger scale, causing many of the though the food is getting, yeah, in a quantity, a large quantity, it is not getting destroyed by the pest. There is economical development also. No much financial resources are being lost. Everything is being saved, but the increased use of these pesticides on the food materials, that is causing other health problem for the human beings. So there is a long-term side effect on the agricultural products by the use of these pesticides or the insecticides. Now going towards the toxicity of insecticides, especially the organochlorine compounds. Uh, this one we are actually mentioning like nowadays we can hear a lot conditions of suicide and homicides using these insecticides. So how to manage that immediately in case because these are all when we learned in the last chapter or the last class we told that it should be less toxic whichever insecticide or the disinfectant which we are using, it should be less toxic for the people who are using it or to the human health. At that time itself, I told you it is not possible. It is somehow some toxic to us also. It's not that we are going to die immediately the very next second after we are getting inhaled with these poisons, but it is toxic actually. So there are people who use it, especially by ingestion, and then it can result in their death. So the organochlorine compounds, we learned that it is nerve poisons. Initially, they causes a paralysis, then excitability or convulsions, and finally the death of the person. And mostly people use the insecticides for homicide or suicide will be DDT, HCH because it's all very easily available, dieldrin, etc. And if that poisoning is happening, the best method to be used is barbiturate as an antidote to the organochlorine compounds, especially phenobarbital. Stomach washouts are generally necessary. Purgatory may be useful, but don't use any oils or fats. So this will be mostly like a MCQ, which will be the question may be like the best antidote for a organochlorine compound toxicity. So barbiturates being the answer. And in case of organophosphorus, here also the organophosphorus like malathion, phenethion, etc. we explained. Here it is interfering with the mechanism of transmission of nerve impulses. So when a human being is going to consume that, the poison effect will be including the symptoms of headache, giddiness, apprehension, restlessness, cold sweating, salivation, uncontrolled urination and defecation, unconsciousness, and in very extreme cases, even resulting in paralysis of respiratory center and leading even to death. And the treatment will be atropine, the use of atropine. So in case of organochlorine, it was barbiturate, especially phenobarbital. And if it is organophosphorus, the antidote will be atropine. And this is a book that is the technical report series from WHO, where it mentions that in case of various types of insecticides, what is the treatment to be used for poisoning. So you can go through it for your information. For your purpose, our study purpose, I put only two important things. Uh, these two slides, nothing else. Then we go towards the individual insect control. So we already learned in the initial slide all the organisms or the insects belonging to the arthropod group which are producing different types of diseases. So we'll start with each one. How we learn the general method, generally the environmental, the genetic, the biological one. We learned a little more about the chemical one because that is more economically feasible, as, at least for the developing countries or the low and middle income countries. And now we'll go through each particular organism and how we can control it. So starting with mosquitoes because they are the biggest menace. Many of the diseases, many of the vector-borne diseases are caused by the, especially the four species of mosquitoes. So starting with mosquitoes, mosquitoes is constitute the most important single family of insects from the standpoint of human health. They are found all over the world, and the most four important species will be belonging to Anopheles, Culex, Aedes, and also Mansonia. So we'll see a little bit about the life history of mosquitoes, and it goes through four stages. Almost all the insects go through these four stages. So in case of mosquito, it starts with egg, then it will move towards larva, the pupa, and then the adult stage. So the egg stage, usually it will be in accumulation of water. So eggs are laid on the surface of water. 100 to 250 eggs are laid at a time. And if it is a favorable condition, like enough water, temperature, humidity, everything is there, the egg will last for about, that stage will be for 1 to 2 days. And after 1 to 2 days, this egg will be hatching out to the larval stage, which is actually the free swimming creature with an elongated body. And that stage will be for about 5 to 7 days. Then, then it will go towards the pupal stage, which is actually the resting stage of the mosquito. It does not feed at that time, but prefers to stay quiet at the water surface. But when disturbed, it will be rapidly swimming towards the downward, and this pupal stage lasts for 1 to 2 days. And in case of adult, after the pupal stage, the pupal skin or the chitinous layer that will be splitting, and the adult mosquito, what we call as imago, that will be emerging out. One second. Yes. Uh, and then, yeah, in case of adult, the adult comes out, and that will be for 7 to 10 days. And normally, the adult mosquito lives for about 2 weeks. And in this 2 weeks, when we see, it is only 2 weeks they are living, but in the 2 weeks, they create all the menace. So 2 weeks only, 14 days only, but they create all the menace in those 2 weeks, and the males are generally short-lived. And now, why we are learning? If you are thinking like, why Dr. Mira is explaining all these X stage, larval stage, pupil, and adult, because now when we go, how we are going to kill or control these organisms, we will be classifying it under how we are going to deal it in a larvicidal stage, when how to kill the larva, and then we'll be seeing how we can, because the pupil stage is not possible, they are not ingesting anything, it will only the contact poisons will be working at that time. Then when it has grown into an adult stage, how we are going to control it. So that is why we need to know, and in this all the three stages, egg, larva, and pupa, you see from the habitat, they are actually in happening within the water surface. So if we are going to control the mosquitoes in the three stages, the egg, larva, and pupa, we actually need to deal with the water where it is being hatched out. So that is why we are learning what are the stages and where we are finding, and also the days also. So how frequently we need to repeat this, the controlling measures. Only if we know the life cycle, the duration, we can apply those. So these are the stages: the egg stage, the larval stage, the pupal, and the adult stage. Now, when we go to the mosquito control, as I already told, we are going to classify into the measures, the larval measure, anti-larval measures, the anti-adult measures, and protection against mosquito bites. So in the anti-larval measures, the first one will be the environmental control, where we are going to do the source reduction, or the breeding areas of the mosquitoes. We are going to control the breeding in that stage. So we can do environmental control over there. Chemical control can be done. We saw that abate is more of a good larvicide than even biological control. The biological control is actually the guppy, the fishes that we, gambusia that we told. So they are not going to kill the adult mosquito because it is in the water. So what they can maximum do is to kill this mosquito larva. So they are actually in the larval, anti-larval measures, or the larvicides. And if it is the anti-adult measures in the outdoor or indoors, we need to go for the spraying method. The residual sprays, the space spray. Residual sprays will be within, that is happening indoor within the buildings. And space spray, like if we have to cover a long area, a large area, the space sprays can be done. And then the genetic control. But as we already told, the genetic control, it's still on the process only. But this genetic control is not done in the larvae, but in the adults. And then if at all this is not, the third possible is how we can prevent ourselves. Mosquitoes are there, but how we can prevent ourselves from getting bitten. So we can use the mosquito net, the window screening, the door screening, and also certain repellents which are safe on the skin that could be used. So going detailed, the environmental control, which is the permanent solution, or the source reduction, where we are going to control the breeding places. So source reduction also implies rendering the water, because it is, we already saw the three stages. The egg, larva, pupa is in actually the water. So we are going to render the water unsuitable for the mosquito breeding. As for example, changing the salinity of water or some chemicals being used. So for this, we need to know the breeding habits of mosquito. And luckily, the four mosquito species that we learn, all of these, they have specific affinity for special type of breeding places. Like all of these are seen in water itself, but the type of water, the water pool in which they are seen, it is very species specific. So the Culex mosquito, one of the most important parasitic for the filariasis spread, usually they are seen in dirty water, in sewage, wastewater, septic tank, if the water is leaking from the, so it is in the dirty ditch water the Culex mosquitoes are seen. So we, if it is the filariasis that is more prominent in a particular area, we should make sure that we are killing the mosquitoes and there is no collection of water, the dirty sewage water in the neighborhood area, because that is the ideal breeding place for Culex mosquito. And if it is Aedes mosquito, mainly for the yellow fever, dengue, etc., it is usually seen in artificial water collection things, like in our house surroundings we may be throwing out certain broken mm, uh, the vessels, containers, bottles, etc., even coconut shells, empty pots. So water being collected on these artificial collections of water in this area, the discarded tins, etc., it is where the Aedes mosquito, their breeding places. So if it is the dengue fever, yellow fever, etc., that we have to control the Aedes mosquito, means their breeding place is actually in this artificial water collection, the coconut shells or the broken bottles, whatever. So that has to be removed. Or we should make sure that there shouldn't be any empty pots, and also it is always kept dry so that the water is not available, or just discard it properly. And if it is Anopheles mosquito, the one which spread malaria, it is usually seen in clean water. Clean water. So what we have to make sure that the water is not exposed. Like if there is the drinking water that is being kept openly, we should make sure, because this clean water is the particular breeding area for the Anopheles mosquito. So we should make sure that it is being properly covered so that Anopheles mosquitoes are not getting bred in those area. And finally, the Mansonia mosquito. Mansonia mosquito, actually, it is not on that particular water they are depending upon, but all the three stages until they become adult, they actually need some aquatic plants like pistia. And it is on that pistia plant, its leaf and also in the root part, they get so attached in the early initial stages. So if we have to remove this Mansonia, or to control its breeding, the best ideal thing is to remove those aquatic plants. So if that aquatic plants is removed, they even then they won't be able to attach with those plants and they are not going to survive. So better we remove these aquatic plants using some herbicides or manually destroy, Mansonia can be controlled. So this is how we need to know the breeding area and what we need to do. So in case of, once again, with Culex, it is in the dirty water, the septic tank, the sewage water where we see Culex. In case of Aedes, it is in the artificial water collections, we see Aedes. In case of Anopheles, we see it in clean water. So better not to keep any water that is being exposed. And then Mansonia, because it will be seen attached to that particular Pistia plant, remove that plant. So this is a plant we usually see in case of like people who are growing fish, fishing and all. So this is an ideal place for Mansonia to grow. But then in this water itself, if we are going to put Gambusia or the guppy, then it is very ideal because they will be eating the larva. Now, going for the chemical control for the larvicides actually. So we can use mineral oil, Paris green, and synthetic insecticides. Mineral oil, it's actually very easily done by each household actually. Whenever there is a drainage in front of us and the mosquitoes are breeding, we can put just some mineral oil or even kerosene, and that will produce a film in that water area, and that film will prevent the oxygen or the air to get into the organism, the larvae, and that can kill the organism. So this is something the household method that we use generally. So the mineral oil, it is the oldest known mosquito control measure. We can use diesel, the fuel oil, kerosene, etc. Nowadays we get mosquito larvicidal oil, the special or specific oils, and this will kill the larvae and pupae within a short time. What they do is, as I told, they will be producing a thin film, and this will cut off the air supply, and that will kill the larvae and pupae. Since the life cycle of a mosquito occupies about 8 days, so we need to apply this once in a week in all the breeding places. But one of the disadvantages, it will make the water unfit for drinking, and it can kill fish. The second one is Paris green or copper acetoarsenite. Remember the chemical term for the Paris green. It is an emerald green microcrystalline powder, and it is actually having the poison arsenic oxide in 50 percentage, and it is actually a stomach poison. So this is not a contact poison. When we are applying, we should make sure that this larvicide is somehow being eaten by the larvae. The pupa is not going to eat anything because it is in the resting stage. The egg is also not going to have it. But then this is the larvicide. So it is the larvae that have to eat because it is a stomach poison. And then only Paris green is going to act. And this is especially good to kill the Anopheles mosquito because Anopheles mosquito are actually surface feeders. So they will come and eat the food materials or the whatever that is appearing on the surface. So this is more ideal to kill the Anopheles mosquitoes. And Anopheles, we already learned that it is seen in the clean water. So if it is applied in the dosage that is mentioned, then this is not going to harm fish, man, or even domestic animals. So it is somewhat safer to use. But the action is restricted. Action is restricted means it has to be ingested, and only the Anopheles is ideal because they are the surface feeders. The last one, the synthetic insecticides, that is the fenthion, chlorpyrifos, and abate, the most effective larvicides. If you remember, just now I told you that it was given to me to keep or put it in the water accumulated areas in case in future. So abate, the concentration that we have to use is 1 parts per million of water, and it is very effective larvicide and also the least toxic. The organochlorine compounds, that is the DDT and HCH. Abate is actually organophosphorus. The organochlorine, DDT and HCH is not ideal to use because their long residual effect. So that can result in a water pollution, water contamination, and increased risk of developing resistance in the vector mosquitoes. So we don't use that organochlorine compounds. And going for the larval control, the biological control, which we already mentioned, the gambusia, the mosquito fish, or Lebister reticulatus or guppy, that can be used for the control of the biological control of larvae. These are the fishes. So these fishes can be used in burrow pits, sewage oxidation ponds. What is sewage oxidation ponds? We will be studying in coming up chapters. The ornamental pond, cistern, farm ponds, etc. So wherever there is a water accumulation, we can use it. However, it is recognized that biological control can be effective only when used in conjunction with other methods. We go towards the anti-adult measure where we use the residual sprays. So for the residual sprays, it is mostly to control the insects inside. So we will be spraying inside the house. DDT is the most ideal one, and the dosage that is used is 1 to 2 gram of pure DDT per square meter. You don't have to remember that. And also to be applied 1 to 3 times a year to walls and other surfaces where mosquitoes rest. And in areas where DDT resistance is encountered, we can go for malathion and propoxa or OMS 33, and to a lesser extent, gamma XCH or lindane. Uh, so that is used for the indoor spraying, and we call it as IRS or indoor residual spraying. Mostly done in most of the African countries because malaria is so endemic over there. Then we go for the space sprays when it is to cover a large area or the insecticidal formulation is sprayed from the atmosphere. So it will be coming out in the form of more mist or fog and to kill the insects. The most one commonly used one is pyrethrum extract, which is taken from the pyrethrum flowers. We have seen that it is a contact one and no poison and also immediately killing the organism. For domestic purpose, the handgun with a fine nozzle that is necessary, but in a large scale, we need to use power sprayers. Uh, Pyrethrum sprays are effective. It will be immediately killing the mosquitoes, but this reduction is only temporary because there is no residual action. That is why we saw earlier, usually pyrethrum is mixed with DDT and to be used. Then, so this is not the space sprays. The first one or the to be considered is pyrethrum, but they are not having residual action. But then we can use for the residual insecticides for the space spraying, especially malathion, phenetrathion for ULV or ultra low volume space spraying. So those new equipments are being used for the space spraying. So one is the conventional sprayer, another one is the thermal fogging, and this one is the ULV fogging. So from this picture, you can somewhat assume how this aerosol or the mist is going to affect the mosquito. In the case of conventional sprayer, the droplets that are produced will be larger in size, and hence it won't be much effective to kill the mosquitoes in a large scale. When compared to that, and this is also going to, the droplet size will be larger, and also this is going to take a lot of time, a lot of labor will be required, manual labor. That is with the conventional sprayer. And in case of thermal fogging, this is coming in a smaller size, yes, but this is not ideal to be used in a indoor one because it will be leaving a lot of um, residual gases. So it is not ideal to use indoor. But ULV fogging, we can use it for space spraying and also for the indoor one. So you can see the droplet size being very ideal, and it is not going to leave any exhaust gas for a longer time. So even ideal for the indoor usage. So it is the ULV fogging which is very ideally used nowadays. This is the space spraying where they need a special type of instrument, the nozzle, etc. Now the genetic control, we saw the different methods, the sterile male technique, cytoplasmic incompatibility, chromosomal translocation, gene replacement, sex distortion, etc. But everything being in the research phase. They have advantages being cheaper and more efficient, but only if it is being widely, we are able to produce it, not in the research phase. Research phase is of course going to be costly, but once it is being introduced, it will be running for a longer time. So then it can be considered as a cheaper one. And above all, not subject to any vector resistance that can be avoided, but still it's only in the research phase. So we learned the anti-larval measures to control the mosquito, the anti-adult measures, and then we go how to protect ourselves from the mosquito bites. The best method is using mosquito net. So it should be white in color, the material that is to be used. so that we can identify mosquitoes very easily. There shouldn't be any single hole, and there is a size for the hole that is present in the mosquito nets. It should not exceed 0.0475 inch. I'm not going to ask you that question. Then we can use the screeners, the window and also the door screeners, especially with copper or bronze. And that also shouldn't be, it is the same size actually, it shouldn't be more than 0.0475 inches. This is costly but gives excellent results. Now we can use the repellents over to our skin, and mostly used will be DEET or diethyl toluamide, and that is important, and that is used as a outstanding all-purpose repellent. There are also others which can be used highly effective: indolone, dimethyl phthalate, dimethyl carbate, ethyl oxynediol, etc. Repellents, oculissifuges, are used mainly for the application on the skin, and their chief advantage is the short duration of protection. We go to the next organism, that is the housefly. So housefly, this is the commonest and most familiar of insects which live close to a man, and they are very abundant all throughout the year. The most important one is Musca domestica, Musca vicinia, Musca nebulo, and Musca sorbens. Remember at least Musca domestica. They are non-biting in nature, but they are very irritating, and this is a sign of insanitation. Remember this sentence. This is important for MCQ, because if a housefly is present, it is considered as a sign of insanitation, or the number is as act as an index of insanitation. So when a house is having a large, not even large, even one insect or housefly is there in the uh, kitchen, it's so irritating, even if it is just one only present. So if a larger is present, means it shows how much the level of insanitation in that particular area. So it is not the mosquito. Mosquitoes are menace. They are causing a lot of diseases. Even houseflies causes a lot of GI diseases. We'll see in the next slide. But still, it is the houseflies which are considered as the sign of insanitation. And here also, they also undergo all the stages as like that of a mosquito: the egg, larva, pupa, and adult stage. So in case of houseflies, we saw their breeding habits. I already explained it will be mostly in the garbage, the refuse, the waste materials that they are going to breed. So in the in the order of importance, the breeding area will be the fresh house manure or manure of other animals, the human excreta, garbage, decaying fruits and vegetables, rubbish dumps containing organic matter, and ground where liquid waste are spilled. The feeding habits, actually the housefly, it does not bite. It does not bite as like the mosquitoes. But how they are causing problem for that is because of their feeding habits. It cannot eat the solid food. It will come and sit on the exposed food materials, and they are not able to eat the solid food materials. They need to liquefy it. To liquefy it, what they do is it will vomit on that solid food, and it make a solution of it and sucks in that solid state. And when it is vomiting, that vomit will be containing a lot of the disease causing agents which is present inside the body. So these adult flies delight in sputum, feces, discharge from wounds, open sores, etc. So they are going to vomit something and then convert that solid food into a liquid form, and then going to suck it. And this vomit is actually the one which is going to release a lot of disease causing agents into our exposed food, and that thereby causing diseases. Then, so that it is actually a vomit drop. The fly vomits frequently. This is often a culture of disease agents. Then they are highly restless. The houseflies, this is a restless insect which move back and forth between food and also filth or the feces. So that help in the spread of infection mechanically. Then defecation. They also like the vomiting they do frequently. Another important habit of houseflies is actually they defecate a lot or constantly. So that also acts as a uh, method by which bacteria is being deposited on the exposed food. Then dispersal. These normal houseflies remain close to their breeding places, but they can disperse frequently up to 4 miles and sometimes even more than that from their point of origin. So it is not, in case of mosquitoes, it is their breeding which is close to our human dwellings, and also their biting nature which is causing the disease. But in case of the houseflies, the breeding, of course, if we are not going to keep the surroundings uh, free from all the solid waste, the excreta, the garbages, the spoiled food, etc. That is the breeding places. And then because of their specific feeding nature, the vomit nature, the constant defecation nature, their restlessness, they're going to spread the diseases. So the transmission, as I told, vomit and defecation, I don't have to explain you the mechanical transmission, because when we already saw that they are very restless, they will be moving from the foot to the filth or foot to the feces. And when they are moving, their wings and also their feet, they will be transporting the organisms, microorganism over, because if it is going and sitting on some exposed human excreta, and that is from a human being, a patient who was or who is having certain worm infestations, the protozoal disease or parasitic infestations. We know, I mean, you have already learned that all these parasitic infestations, they will be excreting that ova or the cyst via the stool. So when these houseflies go and sit on that exposed human excreta or animal excreta, this ova and cyst will be getting attached to their feet and hairy legs. And then they go and sit in the exposed foot, and therefore it is called as the porters of infection. The vomit drop and defecation I told you. So how to control them? So studying once again the environmental control, eliminate their breeding places. So here it will be all that waste materials that we have to remove, not the water properly. It is not the water, but the overall improvement in the environmental sanitation, like storing garbage, but not for long, kitchen waste, other refuse bins with tight lids, and it shouldn't be kept for a long time pending for disposal. It should be removed. Efficient collection, removal and disposal of the refuse by incineration, composting, or sanitary landfill. Incineration, we already learned it is burning the things, and composting and sanitary landfill we'll do in the next class possibly. Next class I'm taking water, and after to that it will become. Then the provision of sanitary latrines so that the human excreta will not be exposed in open. So we need to have sanitary latrines, septic tanks, water seal latrines, and sanitary system. Stop open air defecation, sanitary disposal of even animal excreta, stepping up or scaling up the general sanitation. A clean house with clean surroundings is the best answer to the fly problem. The insecticidal control, we can use the residual sprays. So this Musca domestica, it is resistant to DDT since 1948. Okay, so better we can go for other organochlorine compounds, organophosphorus and carbamate. Susceptible flies, if there is, we can use DDT 5%, and so many things are being given with their percentage and the concentration to be used. I don't want you to study all this by heart, but these are the chemicals that could be used. When writing for the exam, just write, I don't want the percentage, but at least a few examples. One or two examples will do. The addition of sugar to insecticidal formulation enhances their effectiveness. Then we can go for baits actually, which is actually containing 1 or 2 percentage of diazinon, malathion, dichlorovos, etc. The cheapest bait is one that is made by mixing 3 teaspoons of commercial formalin with 1 pint of water or milk, to which little sugar is added. So baits can be, I don't exactly remember how it is being prepared, but baits can be. Then certain cords and ribbons can be used, The coats or strips or ribbons which are impregnated with certain chemicals that can be used. These are actually hung like festoons from the ceilings, and the period of effectiveness ranges from 1 to 6 months. We can also use space sprays where pyrethrin, DDT, and HCH can be used. Uh, and in case of this, we might need the repeated applications. Then we can also use larvicides. In particular, the concentration has been given to be used in the fly breeding places. We can use some fly papers like the baits and the coats and ribbons we told. Similarly, some fly papers can be used. And this is actually not any chemicals, but some natural things that is being used. That is mixing 2 pounds of resin with 1 pint of castor oil, and which should be heated together until the mixture resemble molasses. And then with a paint or a paintbrush, this should be applied on a fly paper. And then this adhesive mixture can be, I mean, the fly paper can be hung up in places where flies are seen about. There may be a slight reduction of flies, but no lasting benefits. So in case of the other two, the fly baits and also the coats and ribbons, it was actually the chemicals that being used. But for household purpose, if we don't want to use these chemicals because we are also inhaling that or we may be also touching that. So in that case, we can go for the fly papers where we are using resin and castor oil, and that is being used, but it is not giving any long lasting effects. Protection against flies, even the screening. Screening can also help to give protection against the flies. And as we already told, if it is environmental sanitation that is required, health education is the most important component, because to achieve fly control, we need the cooperation of the people. It requires organized individual and also community effort. Then going towards the third one, the sandflies. Sandflies, if you see, they are very smaller than mosquitoes, and its scientific name is Phlebotomus. Phlebotomus argentipus, Phlebotomus papatesi, and Phlebotomus sergeanti. These are the main important ones. Even if you don't remember these scientific names, it is so fine. But at least remember the sandflies, otherwise called as Phlebotomus species. Then here also the larval stages going through the four similar stages, the egg, larva, pupa, and adult. The disease transmitted is the phlebotomus, which usually causes the kala azar. It is going species wise. I don't want you to remember this specifically, but remember the phlebotomus or the sandflies. It is causing the disease, the kala azar, the sandfly fever, or the oriental sore. This their habits. It is usually a nocturnal pest. Their bite is very irritating and painful, while their presence is scarcely observed, because we told that it is smaller than the mosquitoes. So the bite will be quite irritating, but the presence is scarcely noticed. Usually, what they do is it will be coming out from its place of habit or habitat only at the night, because it is a nocturnal pest. So from the area where they are living, and usually they are living in the crevices of walls, halls in tree, even the halls on the walls inside a room, the dark rooms, the stable where the animals are kept, the storerooms. So it will be there during the morning period, and only at night they will be coming out of it. It's only the female that is biting. And one of the thing of the sandflies is it is incapable of flying over long distance. The other two, what we mentioned, the mosquitoes and also the house flies, they can actually fly a longer distance. But when in case of sandflies, this is usually confined to a smaller area, that is it's usually confined within 50 yards of the breeding place. So why you need to remember that? So when we are going to do the control measures, it is saying that in an area where Kala Azar is actually endemic, we can we can effectively control that this sand flies in a smaller area if we are able to confirm, because it is not going to a wider area, so we don't have to get bothered the controlling measures to in a large scale. But if we do the effective controlling measure within that smaller area where this organism will be seen, that itself is effective to kill the organism. So control of sandflies, the same insecticides. We can use DDT or lindane which is found effective in reducing the sandflies. DDT residue may remain effective for a period of 1 to 2 years, because we have already seen it is like 18 months the residual effect, and lindane only for a period of 3 months. Lindane is actually the BHC or the HCH. So spraying should be done in the human dwellings and also cattle shed. In sanitation measures such as removal of shrub, vegetation within 50 yards of human dwellings, because it is not going beyond that area, filling up cracks, crevices in the walls, the floors, and also keeping the sanitation high up in the cattle shed, poultry houses, etc., should be given importance to control the sandflies. So, as I already told in the class initially, the question may be like, what is Kala-Azar, the causative factor, the signs and symptoms, and how to prevent. So it is then you have to come this control of sandflies and then mention this. Now we go towards, I think I'm not going to finish the chapter, but let's see. The next one is CC flies, CC flies or Glossina. So always remember the other names also. In case of sandflies, the other name was the phlebotomus, and CC flies, it is the Glossina. Uh, they are the blood sucking flies, and it is having a resemblance to the common housefly. The CC flies, it is usually found only in the African continent, and there is a belt or a region where this is commonly seen. So the regions that is infested with CC flies are called as fly belt. The CC flies live less than 100 days, and 4 species are there, and that's not much important. The first 2 species of that is Palpalis, Glossina palpalis and Glossina tachinoides, they are called as riverine species because they inhabit the woodland vegetation near the watercourses. And the other two, the latter two, the Glossina morsitans and Glossina pallidips, it is called as savanna species because they prefer to live and breed in the woodland vegetation in the savanna country. And why we are learning, I mean, I'm not going to ask you, but just understand in case of CC flies, there are 2 species that is being classified under riverine. So these are living, their habitat is near the woodland vegetation near to the river sources, and the savanna in the woodland vegetation near to the savanna area. So when we do the controlling measures, it is according to their breeding place that we have to concentrate the controlling. So it can attack any organism, man, animal, wild animals. They have used the term wild game. It's actually wild animals of all kinds, birds, lizards, anything they can bite, and both the sexes actually bite. The riverine species is actually having distinct attraction to man, and there is a special feature of this CC flies. CC flies actually they very rarely enter houses, but they are actually they can follow the travelers both by road and rail for considerable distance in order to obtain a blood meal. So that is the importance. They are not entering into certain houses and then causing the thing, but they are actually following the travelers via road or rail. So when we are doing the controlling, the control measures shouldn't be within the indoor or within the houses. In case of CC flies, we should remember they have such a particular nature following the travelers by road or by rail. That means it can travel. It is not flying, but it is via the road or rail. It is traveling for a longer distance and then causing the disease. So when we are going for the controlling measures, we should concentrate in case of CC flies in this method also, or this transportation method. So they are causing the trypanosomiasis or the sleeping sickness. which can affect man and also domestic animals. And control, we can use the insecticides. So Glossina have not yet produced any resistance to insecticides. We can use DDT, dieldrin, for controlling the CC fly. Insecticide is applied from aircraft when large areas are to be covered quickly, because I told you they are not flying by themselves, but by road or rail they are following travelers, which means if we are to cover a large area, then better go for applying for a space spraying. Then clearing of vegetation where the CC flies are living to control it. But usually this is going to give a good result only when we are going to use the insecticides also. Then there has been game destruction. Game destruction was like, because it is affecting all the wild animals. At earlier times, we saw that sisifly is usually confined in the African continent. So to kill or to clear these sisiflies earlier, large forest area in the Africa was cleared, killing even the wild animals. But because of the environmental catastrophe that is being caused, this method is now given up. Then the last one is genetic control. Where, they are especially using the sterile male technique to control the CC flies population. Okay. One of the next one is black flies, and this is also called as Similidae. They are small, robust flies and almost black in color. It can attack domestic animals and also man. They are causing the disease onchocerciasis, and usually their breeding place is in the water. The eggs are laid on the submerged stones and water weeds. The larvae are aquatic, and they fix themselves to the stones or plants, usually at a depth of 1 foot. So control of the adult fly is difficult, because once they become adult, they have a high range of flying, that is about 100 miles. So the better method to control black flies will be to use a ideal larvicide, because usually they are seen in the water, and better to use in the when they are in the larval stage, because once they become adult, they can travel a large area. So controlling at that level is very difficult, better to control at the larval stage. Abate. Abate is the ideal one to be used, because we have seen that abate, when used in the concentration of 1 parts per million, it is not causing any damage or any harm to human or the fish or other aquatic animals. So abate is used, which can kill the black fly larvae without causing any harmful effect. It is added to river water in weekly doses of 0.05 to 0.1 milligram per liter over a period of 10 minutes, or the 1 ppm. Okay, lights, it is more. So maybe we can stop over here. 20 more slides. Fine. Do you have any doubts? Okay, let I go and stop the recording.